Hello, everyone, and welcome to the first stage seminar in 2024 and the first sunshine day in many, many days. Thank you, Michael. Before introducing today's speaker, I would like to note that the stage seminars operate with the support of CANSI Ontario. CANSI stands for Canadian Statistical Science Institute, and are also sponsored by multiple institutions, including SICKITS, Lohenfeld, Maloglan Center, and the Department of Statistical Science. We're in the Department of Statistical Science seminar room. And today's speaker, Dr. Wu, a professor of biostatistics at the Fred Hutchinson Cancer Center and the University of Washington at Seattle, I think needs no introduction, as I bet most of you, if not all of us, has used or heard about the SCAT, the sequence kernel association test for real variants. And the landmark paper Wu et al. 2011, published in the American Journal of Human Genetics, I checked this morning, has a total citation of 2,585. If you check it now, maybe one or two more. And more recently, Dr. Wu's lab has focused on the studies of human microbiome. And today we're very excited to learn more about how to use statistics to keep our microbiome analysis out of the toilet. Thank you.
All right, thank you very much for the kind introduction. Thank all of you for being here today and for the invitation to enjoy this wonderfully sunny weather. Anybody who's been to Seattle in the winter knows that it's fairly horrible. I've heard that you guys have had a taste of that. One of the first things I do is I buy sad lamps for all of my postdocs who come join us from other places. The fact that I have to do that should tell you about what's going on there. Summer is delightful. That's when we try to do most of our recruiting, and then we trick people into showing up in October. All right, so today I'm going to talk about some work that we've been doing on analysis of the human microbiome, and particularly addressing challenges that come about when we start dealing with what I call large-scale microbiome profiling studies. So you'll have to bear with me. I'm in a room with an audience that knows more than me about everything. There are people who know more than me about the microbiome, as well as people who are statistically much deeper than I am as well. So I'm somebody who sort of dabbles in between, and so I apologize if I say anything too egregiously wrong. All right, so the human microbiome, over the last 10 years has been one of the hottest topics, and it's been fabulously successful in being associated with all sorts of different complex diseases and what mostly are considered exposures. That's the typical way that we oftentimes model the microbiome. And so things like diet, exercise are very strong drivers of microbiome profiles. You have things like drugs, smoking, alcohol, as well as treatments and other types of more classical environmental exposures like air pollution, dust, and such things, all driving microbiome composition. Then there's also things that we think are driven partially by the microbiome, things like asthma, cancer, diabetes, and even whether or not you respond to cancer immunotherapy. We think that all of these are potentially related to your microbiome profiles, and there's been considerable work in all of these areas. One thing that is a little bit different between host genetics or human genetics and the microbiome is that I have to put a little bit of a question mark by outcomes. The reason is, all right, your genetics are probably not driven by your asthma status, but here, because of how samples are taken, unless you have a longitudinal component, it's actually fairly difficult to distinguish whether or not the microbiome is resulting from a change from your differences in health conditions, such as whether or not you have cancer versus the other way around, truly having the microbiome driving tumorigenesis. All right. So it's a little bit messy for those reasons, but nonetheless, we have found many associations. Many of these are robustly identified, but there are also limitations. So let's take a traditional microbiome study. Typically what happened was you would take a very happy, healthy mouse, and you'd take a very, very sick, unhappy mouse, all right? Then you take happy poo from the happy mouse and unhappy poo from the unhappy mouse. And you sequence them and you take a look at the different bacteria that are in there and you compare basically their compositions. What characterized these were they tended to have very, very simple questions and very simple designs. So an exact example of something that happened was some people took some mice. they fed them their own body weight in NutraSweet, and then the other one, they did not. And they said, hey, look, if you feed an organism its own body weight in NutraSweet, their poo looks a little bit different. Seems reasonable. And because of that, they were able to find some associations, all right? They were able to do these with very, very small numbers of individuals and organisms, sometimes 5 rats in a cage, all right, 10 people, 30 people, all right? And partially this was because you're looking for huge effect sizes. If anybody eats their entire body mass in anything, you're likely to see some differences in your microbiome, all right? So you're capturing these huge, huge effects. The problem is that mice are not the same thing as people, all right? No matter what some people seem to think, all right? If mice were the same as people, we would have cured cancer about 50 times over. We would have understood diabetes and cured that as well 50 times over. The reality is that mice experiments are not sufficient for understanding human systems. Humans are way more complicated, all right? Partially, there's a lot more heterogeneity among humans. Fundamentally, you're not allowed to lock people up in cages and feed them their own body mass in NutraSweet. You're not even allowed to do this with trainees and graduate students, all right? It's a joke, by the way. I would never lock my students up in cages. I asked my IRB and they said I wasn't allowed to. And by the way, if you do undergrads, they actually go through the animal care use committee. But you guys are a tough crowd. Anyway, none of that is allowed over here. Basic point, humans, cages, bad, all right? Despite what some people in the CIA seem to think. So the result here is that when we deal with humans, many individual findings actually replicate very, very poorly. And part of this is that when you deal with humans, the effect sizes that you're talking about are simply much more modest, all right? We're very, very different. There's a lot more complexity to humans. And so you need much larger sample sizes in order to truly tease apart the effects that we truly care about over here. You have to deal with much more confounding, all right? And again, you're not feeding anybody their own body mass in anything over here. So this has really motivated the development of what we consider large-scale microbiome profiling studies, all right, where you're now taking large numbers of patients or participants over here. And so what the major advantages here are that you can robustly and powerfully identify particular associations. You now have the sample size now to really do something more sophisticated and pinpoint what's going on. You can ask more sophisticated questions of your data. You can start looking at things like mediation analysis, things like interaction analysis, effect modification, and so on and so forth. You also tend to have very rich, what we consider metadata or extrinsic things to the microbiome, things like exposures, risk factors, outcomes, and other omics data. Many of these studies have been ongoing for a very long time. These are cohorts which we followed for 20, 30, 40, sometimes 50 years. So you have very rich data to go along with that. Examples of these include the CARDIA cohort, which at its initial microbiome profiling study had about 600 people. PIN is a vaginal microbiome profiling study in pregnant women, which has about 1,200. Hispanic Community Health Study had 3,000, MEC 6,000, NCI 10,000, and nurses now, it's well beyond 25,000. I don't remember the exact number. I think somebody today told me like 150,000, I can't even remember. But regardless, it is enormous numbers of participants over here that really allow you to powerfully identify associations with much more reliability and allow you to ask more sophisticated questions of the data. But with these larger sample sizes come additional challenges, all right? Some of these challenges are inherent to large sample sizes. For example, batch effects. So we'll talk more about the details of these, but basically batch effects arise from differential processing or handling of specimens, all right? When you only have 5 rats, you can sequence all 5 of these guys together very quickly. When you now have, 6,000 people, then this needed to be divided up into perhaps 30, 40, 50 sequencing runs, all right? So you now have batch effects from there, not to mention differential handling, patients and participants coming from different sites, and so on and so forth. This is simply resulting from the fact that your sample size is bigger, so there's going to be more going on there. The other thing is that with these larger sample sizes, you have new opportunities, such as the ability to have more powerful association, all right, or to ask more sophisticated questions. And what we found is that there are, first, in some cases, there's a lack of available methods for dealing with some of these questions. And secondly, with your larger sample size, reliable implication of individual bacterial taxa, and by taxa I mean species, genera, so on and so forth, is much more challenging over here. So, and we'll talk a little bit more about that as we go through this presentation. But these are the two challenges that I want to talk about today. How do we deal with batch effects? And secondly, how do we do more robust testing within the context of microbiome profiling studies? So the first part of this, all right, conquering batch effects in microbiome with data with Conquer. Conquer is the name of our method. I did a poll. I asked people, should it be called Conquer or Concur? And people seem to like Conquer better, so hence that's what it was. I actually kind of like Concur because you're trying to get different data sets to concur, but you know, it's a democracy, so that's how it works. All right, so batch effects. As I said, this is systematic variation in the data that arises from differential handling or processing of specimens or samples over here. Some of this can be from different sequencing instruments. It can be machine drift. So when you are sequenced, even if it's on the same machine, all right, how long it's been in storage or how early it was processed. It can be as simple as which batch of reagents did you use over here. Different batches of reagents do make a difference. And as we found actually with the microbiome, they have different contaminants in there sometimes, right? Nothing is truly sterile. And when you deal with bacteria, different batches of reagents can make a huge difference. And as well as who is actually doing the processing and who is actually doing the extractions and so on and so forth. That can make a huge problem. So, you know, the biggest issue here is that it can lead to false positives and false negatives over here. All right, we tend to worry more about false positives, of course. You know, as statisticians, we control type 1 error, but then we worry and, you know, you know, there are also false negatives. And batch effects have been around for a long time. All right, they've been career killing for a number of scientists. We first discovered these very, very prevalently within the context of proteomic studies, where literally people essentially lost their careers because they identified batch effects rather than true signal. All right, so they're not unique to microbiome data. They're probably common to all high throughput data that you see. I think GWAS may be the one sort of exception where you don't see such large effects, partially because your data are coarsened so much and you're dealing with relatively prevalent things. But even for rare genetic variants, you're going to have issues of batching that you have to deal with, right? So how do we usually deal with this? Within all of genomics, there's basically two strategies. One is what I call batch removal, where the idea is you try and get rid of the batch effect from your data up front, and after that, you have a cleaned data set with which you can do everything else. The other thing is what I call batch adjustment over here. And this is what happens when you try to accommodate the batch variable in things such as your association analysis. All right, so you adjust for it as a covariate or with some weighting somehow. All right. I'm going to focus more on batch removal. The reason is batch removal is oftentimes more popular in genomics simply because you end up with a cleaned data set and you can do more with it. All right, it's no longer just association analysis. You can do things like visualization, prediction modeling, and so on and so forth. And it's simply what is more popular over here. So here's the data example that I'm going to show. So it's going to be based off of the CARDIA study, which has about 600 microbiome samples. And so this is a longitudinal study of risk factors of cardiovascular disease. Microbiome data were collected at the year 30 follow-up. So it's a pretty old study. It's been around since the mid-80s. And other covariates that they have include all sorts of things related to cardiovascular disease, blood pressure, demographics, whatever you want, they have it over here. And so these are what we call principal coordinate analysis or PCOA plots of the microbiome data. Also in statistics, we call them multidimensional scaling plots. So each dot over here, I don't even know, well, it doesn't show. Each dot over here corresponds to a separate specimen over here. And the centroids over here correspond to the centroids of different batches. So here, there are 6 different colors corresponding to 6 different batches. So it's like a PCA plot, except we use a nonlinear measure of distance over here. So here it's based off of Bray-Curtis dissimilarity. And then this is, we transform the data using a center log ratio transformation, and then, which is basically just log transforming and centering, followed by Euclidean distance. which is the same thing as just your regular PCA plot over here. But regardless, you can see that there's tremendous separability by different batch in this case. Now, somewhat reassuringly, if you look at each cloud, the overall shape is pretty similar over here. But there are clear differences both with regard to the spread as well as with regard to where they are shifted over here. All right. So very, very substantial batch effects over here. Like number 2 is totally not overlapping with any of the other ones over here. Okay. So how do we do batch correction then? Well, first we ask, can we just borrow ideas from other data types? All right. So by far, the most popular approach for batch correction in all of genomics is a tool called Combat, all right, which essentially is very, very simple. It basically assumes that you have a normally distributed outcome, all right. In this case, they assume that each microbe is normally distributed. Then they regress that on variables that they know. Those are your X's over here, as well as an indicator for batch, all right. And basically, you just regress out and get rid of that batch effect. All right. Now they do it a little bit more complicated. They use some empirical Bayes and some degree of shrinkage, but with large enough sample size, it basically is exactly this over here. Nothing fancy going on. All right. You just regress out the effect of batch. It was a first-year summer project for the first author who developed it, and it's probably now one of the most cited papers in all of biostatistics. They have generalized this to count data, but they place very, very strong distributional assumptions on count data over here, which tend not to work that well for microbiome data. In particular, for microbiome data, our raw data over here are zero-inflated, very over-dispersed counts over here. All right, we have the counts for the number of different reads corresponding to particular bacteria within each sample for each bacterium over here. All right. So there have been a few approaches that have been developed for microbiome data, but they tend to be very, very specific in terms of requiring certain types of controls in spike-in experiments. Some of them require very, very specific designs, such as only case control studies, and oftentimes they're whacking the data with a huge hammer over here that loses a lot of the characteristics of the data. The other thing is the vast majority of these approaches focus on batch adjustment rather than correction. So yes, you can use these, but it's only useful within the context of association analysis. Other tools, such as generating those PCOA plots and multidimensional scaling plots, you can't do, all right? It's still going to be heavily, sometimes it's going to be heavily confounded, obfuscated by your batch variables over here, right? So that's why we developed our proposed approach. We said, can we do a little bit better than this? And we developed Conquer over here. So here's the punchline for the talk. This is the same figure that you saw before. These are the CARDIA data before batch correction. See that there is quite a bit of variability here by batch, all right, both with regard to the variation as well as with regard to where things are centered. This is what happens after we use Conquer. So you see things are lined up. It is not exactly the same because, you don't expect everything to be perfectly the same across batch, but overall, all right, you see that the shapes of everything are similar, the variability is a little bit more similar, and overall things are centered much more closely to each other, right? So before, it's a mess. After, they at least are lining up over here and looking quite a bit better, all right? So let's talk about what Conquer is actually doing. So Conquer stands for conditional quantile regression. And the idea here is that we're going to correct each taxon separately. In particular, we estimate the distribution of the taxon, covariates, and batch, and then we simply remove the batch variable over here. But what makes this different than Combat is that we're going to use quantile regression over here, right? So like I said, it's similar to Combat-seq, which assumes a negative binomial model and then uses a similar procedure, except here we're going to use quantile regression instead. All right. So negative binomial models are great for many types of count data, but not for microbiome data. Okay. So in terms of estimating the quantiles with zero inflation, all right, and over dispersion robustly, we use a two-part model to address the zero inflation, and it's quantile based in the sense that it's robust to and this results in robustness to complex distributions, such as strong deviations from negative binomial or Poisson assumptions. And it also allows us to correct for much higher order effects while doing this. All right. So here's our simple two-part model. All right. So you have so yi is going to be just the abundance of a particular bacteria, a taxon for the ith person over here. And so it's either 0 because, well, this bacteria is not in there, or it's non-zero. And so we model that over here as depending on a bunch of covariates and batch over here. Then for the continuous component, or I should say the count-based component, we jitter it a little bit so that it has to be jittered in order for the fidelity of the model. It's a quantile model, and so if it's discrete, it doesn't really work out very well, right? And then we allow it to depend on, again, the covariates and batch over here. And so the idea behind our approach is very, very simple over here, okay? For each taxon, we fit the two-part quantile regression model. There's a little bit of messiness, which I'm not going to go into great detail on, but we can do it. Then we estimate the original quantile function for each sample. So now this is for each taxon, so for each microbe that we're looking at. And for each sample now, we're going to estimate its quantile function over here. Then we estimate the corrected quantile function of each sample for each taxon by removing the batch, the estimated batch effects over here. Okay. Then we do quantile matching. We locate the original observation and find that it's at the tau hat quantile on the original distribution. And then we pick the corresponding tau hat. Then we go to the corrected quantile function, find the value of Q at tau hat, and that is our corrected value. So just to illustrate what's going on, and we do this for all taxa, just to illustrate what's going on over here. So suppose you have a particular observation. So this is the yi for a particular subject. This is person i's predicted quantile function based on their profile of covariance over here, all right, in batch variable. This is their original quantile function. This is the corrected one after removing the batch effect. So what you do is you see, well, this observation was at about 1500. And so then you come over to here, 1500 is at the 80th percentile over here, 80th, the 0.8 quantile. Then you come down onto the corrected side, you see 0.8, and that maps over here to about 800, all right? So the original value is 1500, the corrected value now is 800. It's just quantile matching in this case, right? In fact, combat and all of these approaches do basically the same thing, except that once you've assumed a normal distribution, it's just easier because you have a couple of parameters that take care of everything, all right? Versus here, we have to make it a little bit more complicated to do the distributional matching. So I'm not going to talk too much about this, but there is some messiness with regard to estimating these quantile functions. The main thing is that you have to condition on the fact that that you can't just use regular quantile regression. You have to condition on the fact that some things are already equal to zero, so that you have a bunch of zero observations. So it's a conditional quantile function. All right. And there's also some challenges with regard to some discontinuities there, which we just linearly interpolate. I'm going to skip some of that. But here's the overall Conquer procedure, all right? We take in all the read counts in all samples. Then for dealing with the zero inflation, we use a simple logistic regression. For the non-zeros, all right, we use a quantile regression model over here. And from there, you can get the conditional distribution. All right, so this reflects the zero inflation, and then this guy over here gives you the quantile regression for the continuous pieces over here. Then over here, you combine these things, you get the conditional quantile the conditional distributions, and then basically we do some degree of matching. In terms of matching, all right, so here's an example from the CARDIA data set. All right, we can actually correct values that are positive, so that are non-zero to 0. We can correct things that are correct things to a positive value from 0 to a 0 value from a positive value, and as we saw before, from a positive to positive value. So we are setting some things equal to 0, and we are taking things that are non-zero and that are 0 and setting them to something that's non-zero. It just depends on how we are on the particular taxon, depends on things going on over there. All right. So rather than dwell on some of the details, let's talk about what actually happened with our data. So finally, after a little bit of cleaning, there's about 380 taxa on 340 samples that were measured across 6 batches. The key variable here that we were interested in was systolic blood pressure, all right, which is a continuous variable. Covariates that we included in our model for simplicity, age, gender, and race over here. Okay, and so these are the PCOA plots, the principal coordinate analysis plots, before and after correction. This is these are the original data over here, looking at Bray-Curtis dissimilarity, and this is using Euclidean distance on CLR transformed data. So again, you see huge batch effects. We saw this already. This is what I showed before. This is what happens if you use Conquer on the Bray-Curtis scale. This is using on the Euclidean scale. Everything looks much better. This is what happens if you use Combat-seq, which operates under a negative binomial model, all right? It's way better than here, but still does not do very well. It looks a little bit better on the CLR transform scale, but again, still does not, at least visually, look very good. All right, so what these plots show is that if I take my data and I whack it with a big hammer, all right, things look very, very similar. Well, the easiest way then to get batch variables to go away is to set all of my data points equal to the exact same thing. All right, there'd be no batch. All right, you have huge bias, low variance. It had no variance either, so it would be fantastic. Low variance, huge bias, but it's something you could do. But this is what we did to show that we still preserve some degree of signal. So the permanova R squared, you can just view it as a multivariate, a particular type of multivariate R squared on these types of nonlinear distances. And it's just the amount of variability that is explained by particular variables. So here, this is the amount of variability explained by batch. All right, this is the amount of variability explained by systolic blood pressure. So in the original data, about 10% of the variability was explained by batch. All right, systolic blood pressure, it's like less than 1% of the variability. All right, this is just how real data are. All right, these variables don't explain a whole lot of variability here. If you use Combat-seq, it does much better. All right, it knocks that down to about 6%, and systolic blood pressure goes down a little bit, but the variability is still largely preserved. If you use Conquer, you see it doesn't set the variability explained by batch to exactly 0. All right, but it's now at 0.05%. Much, much better than 10%, while still preserving the variability that we seem to care about here. So the take home here, it does reduce the batch effect. It mitigates it significantly while preserving the signal over here. So I showed that to my collaborators, and they said, well, you didn't do a random forest. I was like, I don't know why I need to do a random forest, but they said, do a random forest. So we did the same thing. And so we ran a random forest. All right, this is just the cross-validated, probably doubly cross-validated residual mean squared error from random forest using the original data. All right, after applying Combat-seq and after applying Conquer. And you see that, well, if you look over here, the total variability explained by systolic blood pressure over here, the error here is now lower. I don't know what this really tells us that the last slide didn't tell us, except, well, microbiome researchers really like random forests, and if you don't do it, they won't publish it. I don't know, it's a thing, all right? So it has to go into every single grant that we write, like you're gonna do a random forest. All right, I think somebody ran a random forest once, and hence after that, everybody has to run a random forest. It's like just how biology works. Like people find one thing and then everybody has to keep doing it. Don't get it. All right. So here's another quick data example. I'm running a little bit slow. So this is a different situation where what we said was, let's do mega analysis of multiple studies, which is to say we're going to combine a bunch of different studies in order to boost power. The funny thing is in microbiome world, they call this a meta-analysis, even though they're using the original scale data. But we all know in genomics that this is called a mega-analysis, where we're pooling data from different studies to try and gain power over here. So here, what we found in the HIV Reanalysis Consortium is that they have about 10 different studies, which were collected where the collection was under different protocols and designs, and different sequencing protocols, I should say. And so here, what we're going to do is we're going to treat each study as a batch, all right, and try and pool our data together this way. This is much harder than a traditional batch correction. The reason is that even the sequencing protocols and everything that you're doing here is different. So you're really asking a lot from the methodology. But we said, let's just see what happens. So our pooled data, we had about 600 taxa on 570 individuals across 10 studies. All right, each of these 10 studies was considered to be a separate batch, and we wanted to combine them. They ranged in size from 24 to 135 patients. All right, the key variable here in this case was HIV status. It's a simple binary outcome. Covariates included age, BMI, and gender, where gender was a Gamish variable of both sex at birth as well as whether they had MSM status over here. All right, which is, and actually, what we really cared about was some sorts of receptive anal intercourse. We didn't have that, so we were just sort of going with this gender variable in this case. All right. So here are the PCOA plots before and after correction. So you can see before, everything is hugely different. Everything is non-overlapping, all right? Lots of things on the Euclidean scale, looks a little bit better, but still very different. This is what happens if you apply combat-seq. You actually don't see too much change over here. This is what happens if you combat-seq and then we transform. This is what happens if you run conquer. It looks better. It's not perfect, but it looks way better than these guys over here. On the Euclidean scale, you see it does look way better than this. Everything is together, but it is still not perfect over here. If you actually look at the variability explained, all right, so on the original scale, 30% of your variability is just from the batch variable, all right? If you use combat-seq, it drops it down to about 21%. Way better than 30, but still not all that great. If you look at Conquer, you see it knocks it from 30 down to about 1.2% over here. So much, much better is what you would think. Unfortunately, all right, and it is way better, but unfortunately, if you look at the variability explained by HIV status, it is still tiny compared to the variability explained by batch. So Conquer helps, is how we say it, is how we like to say it, but it is by no means a perfect solution. But it's way better than if you choose than not doing it. So if you're going to do association analysis, you might still want to try and adjust for it there, for batch there, but running this can still be helpful for things such as visualization. All right, here's the AUCs from random forests. Again, we had to do that because people wanted to see it. It says basically the same story as what was on this previous slide. So just a couple of remarks for this problem, for this. All right, in general, batch correction for microbiome data, it's increasingly important, both as studies get larger and as we hope to pool larger studies together, all right? Conquer is a general, relatively robust approach for batch correcting microbiome data. It works directly on the count data. It does not make strict distributional assumptions over here. It's quantile based, and it seems to remove the batch effects relatively well while preserving signal of interest. It is, of course, not perfect over here. There's some additional options that I didn't really talk about. We've implemented approaches that include some degree of regularization, and you can also tune models across different ranges of taxon prevalences and do slightly different corrections for, say, more abundant versus lower abundance taxa over here. Here are some of the limitations of Conquer, and actually basically any batch correction approach here. First of all, it doesn't work that well for rare taxa. You just don't have enough data to estimate it, and this is true for any of the batch correction tools that we've used here. It doesn't work very well if your batches are all very, very small. If every single person has their own batch, then there's nothing to do. We just go home, all right? All right, you're sort of screwed, all right? Here, we're also correcting each taxon individually. We're not considering the correlation among taxa when we do this, all right? So it doesn't necessarily preserve correlation in the original data. Now, whether or not you care about that is arguable. Perhaps in neuroimaging data, that's extremely important. But for a very unstructured data, such as what we have here, or where the structure is unknown, trying to capture structure that we don't know oftentimes introduces more variability into the process than what you would say. Statistically, a challenge here is that we have included our variable interest in the model already when we do the batch correction. So in principle, you are double dipping if you then do association analysis afterwards. This is again true for all of the batch removal tools, including combat. All right, theoretically, your standard errors are going to be a little bit lower than what you expect. In practice, however, and in our simulations, we have not seen any increase in our actual false discovery rates. So, it truly is a statistical issue, but it's not one that seems to have any practical impact on deployment of these approaches. And so we've sort of swept that underneath the rug over here and stomped on it, as we are wont to do sometimes because I have data that I need to analyze. Finally, one thing that I wanted to add on here, but I didn't, is that Concord doesn't take into any strict biological information. So if you look at how people do normalizations for other data types, they build in things such as GC content, they build in things that are inherently biological covariates into there. We're not doing any of that right now. That is something that we are looking forward to pursuing going forward. All right, with that, being said, I'm gonna sort of hurry through the second part of my presentation, which is dealing with the opportunities that come about because we have a larger sample size. And one of them is that we can now more robustly, in a loose sense, identify individual taxa that are related to outcomes. So, but here by robust, I just mean more reliably identify them. But it's also gonna be robust in this case, in the sense that we're using quantile-based approaches again. So, association analysis is probably the most common recurring theme in most of microbiome analysis, which is to identify individual taxa associated with variables of interest, exposures, and/or outcomes. This usual approach for screening, and this could actually be true for any high-dimensional data type. You normalize, do some pre-processing, scale your data. For each feature, in this case, each taxon, you assess the association with outcome. You control for multiple testing, use either FDR or family-wise error rate if you're going to do GWAS, right? And then you do all sorts of auxiliary analyses, whether it's pathway-based analysis, network analysis, all sorts of strange and wonderful things that you might want to do on top of the vanilla analysis. So within the context of microbiome analysis, there are two major classes of approaches that are used. The first one is you just apply a classical statistical test. By that, I mean linear regression, Wilcoxon rank sum, all right, whatever you want to do. You just apply a t-test, all right? The other strategy is to use what I call loosely tailored approaches. These try to take into consideration more of the structures that are inherent to microbiome data. And so there are pros and cons to both of these classes of approach. First, what we found is that in general, these classical approaches, such as linear regression, t-test, Wilcoxon test, and so on and so forth, they oftentimes lose power, all right? So they often struggle with zeros and highs, and they don't take full advantage of the data characteristics. All right, so they do a great job of preserving type 1 error, but they are oftentimes not very powerful within these contexts over here, all right? On the other hand, the performance of tailored approaches is even worse, right? The particular choice of normalization dictates which approaches are necessary. So if you're using DEC2, that's on the count scale, versus metagenome-seq assumes some degree of scaling to some sort of cumulative sum medians over here, so it becomes continuous, all right? And so what characterizes these is that they have very poor type 1 error control. And so as statisticians, number one is protect type 1 error, then you worry about power. These guys over here don't even protect your type 1 error. Unfortunately, they are also probably the most popular approaches used within the field over here. So we knew approaches really are necessary. We're not the only ones to observe inflated type 1 error of these tailored approaches. This is commonly known over here. There have been tons of papers that have shown these. One of the fundamental issues of these tailored approaches is that they're all highly parametric in requiring a particular distribution. So DEC2, EDGE-R were developed for gene expression analysis, and they make a very, very strong negative binomial assumption here on the distributions. Metagenome-seq it assumes a zero-inflated log Gaussian or Gaussian. There's tons of other zero-inflated approaches, zero-inflated gammas, betas, negative binomials, Poissons, whatever you want. There's all sorts of count models and all sorts of things that people have been developed. The problem here is oftentimes that these distributions don't really match the true distributions. So what you're rejecting is not that there's a difference in the parameter values, but what you're rejecting is the fit of the model to this distribution in the first place. Now, if you only have 5 samples, all right, there's no way you can tell that something is not negative binomial, all right? But now when you have 5,000, those deviations from negative binomials suddenly become apparent over here. All right, so it's as much something that comes about because of large sample size and the opportunities, as well as illustrating the deficiencies the dangers once you have larger sample size. Again, 3 samples, you can't tell me it's not normal. You can't tell me anything about it distribution-wise. And so if there's a huge effect, all right, I'm going to find it. All right, but once the sample size gets large, it becomes a problem, these modest deviations over here. So this motivated us to develop what we call the zinc method, a zero-inflated quantile model over here. And so what we do is we achieve simultaneous conditional quantile inference for zero-inflated abundance over here, where we're basically going to be like the Wilcoxon test and many of the other non-parametric tests here. We're basically testing for equalities of distributions now. All right, are the distribution of my cases the same as my distribution for my controls? Very, very simple over here. All right, so as I said, it's a distribution test. What we're going to do is we're going to summarize the difference at the 0 counts over here, and then add a grid of quantiles for the non-zero component. All right, so, and then we're going to look over both the 0 component and this grid of quantiles and see if there's a difference. Operationally, we use a rank score-based test for the quantiles, which makes it computationally efficient, even if you have relatively large data sets over here. The main advantages here, it's robust in the sense of limited distributional assumptions over here. It allows for covariate adjustment, and it allows for detection of heterogeneous effects across quantiles, which I will talk about when we get to some of the results over here. So again, this is our 2-part model. Looks very, very similar to before, except we no longer have a batch variable over here. All right, so logit for 0 versus non-zero, and then this is for the continuous or non-zero component over here. So this characterizes the effect of covariates of interest, and here is your clinical covariate of interest, and we're interested in testing whether this coefficient function over here is equal to 0 or not. All right. So to test this, all right, very, very straightforward. All right. So we test whether this guy is equal to 0 for all of these tau values versus otherwise over here. Okay. The testing strategy over here is very straightforward. For testing the 0 components, we just use a simple logistic model. All right. 0 versus non-zero, you use a logistic regression. You can use any sorts of your usual tests that you would use in this component. Then for the quantile-based tests, we test across a grid of quantiles, all right? But then when you're doing this, you have to account for the bias from the fact that you're only using the positive part. You can't just fit a usual quantile regression. It has to be conditioned on the fact that you have a bunch of 0 values over there that are shifting it already. Then once we have this list of p-values for the logistic component, as well as a grid of these k different p-values for the non-zero component, all right, we just combine the p-values over here. All right, and there's a couple different strategies that one can use for doing this. So this is an overview of the approach. All right, basically, all right, you basically take the 0 versus non-zero. This goes into a logistic model, and then for the continuous component, it goes into a quantile-based model over here. So you test for whether this guy is equal to 0 or not using your standard logistic regression tests, 1 degree of freedom test. And you can do a series of quantile rank score tests over here in order to assess the association at each choice of quantile. Then in terms of combining all of these p-values, we either take a min-p approach, which requires some more tuning in order to deal with correlation, or we use a Cauchy combination test, which is one of the easiest and most simplest things that you can apply these days for averaging the p-values. Okay. So here's the rank score adjusted for 0 inflation at the tau quantile. So this is basically looks a lot like your usual rank score test, except here we deal with things conditional on being non-zero over here. All right, and so some adjustments are necessary in order to do this, but nothing fancy over here. All of this is pretty straightforward, right? And so to do this, we do have to do some work in order to get the correlation to work out. But again, it works out. We can do it. In terms of combining the p-values, you can use a min-p approach. All right. So basically, you take the smallest p-value, let that index significance, and then you do some degree of resampling in order to accommodate the fact that you took the smallest p-value. Alternatively, you use a Cauchy combination test. All right. So you take your p-values, you use your probability interval transform, shift them to a standard Cauchy, you average on the Cauchy in the Cauchy sense, and then you find the tail probability over there. Remarkable fact is that when you average Cauchys over here, that it's very, very robust to the correlation over here, particularly in the tails. So at least your tail probabilities and p-values seem to work out just fine. All right. So here, we're going to revisit the CARDIA data. Hide this. All right. So the outcome in this case, it's going to be either the normalized taxon abundances. We're going to consider rarefaction as well as cumulative sum scaling, just to consider two different scales. One is discrete, counts, rarefaction. The other one is continuous over here. All right, depending on the choice of normalization that you use. Covariates we care about: age, physical activity score, a dietary score, and race that we adjusted for. And the question is, we were looking at the genus level, and we wanted to understand which genera are associated with hypertension. In this case, high blood pressure versus non-high pressure. If we set the false discovery rate to 0.05. We first conducted some simulations looking at type 1 error. We have tons of these, but here are just some examples. All right, so in this case, we just permuted covariates, and then we created 50 null data sets. And you can see the false discovery rates over here across different data sets. So this is corn-cob, which assumes a beta binomial distribution, DEC2, which assumes negative binomial, EDGAR, negative binomial. The linear LDM is called a linear decomposition model. It doesn't make any assumptions, but uses permutation for inference. Limma is commonly used for gene expression data. Linear regression, this is monocle, an approach used for single cell sequencing. And QRank is a quantile-based approach that does not accommodate zero inflation. And this is zinc using a Cauchy combination and zinc using the min-p. You can see that most of these approaches that make strong distributional assumptions all have inflated type 1 error, all right? It's simply because your real data don't match those distributions, right? So even though there's plenty of people using DEC2 and corn-cob and these things, we think that that's oftentimes, well, you're just gonna have a lot more false positives than you expect. On the cumulative sum scaling data, different methods apply because these are now continuous. So the LDM linear regression, both seem okay overall. Metagenome-seq, which assumes a zero-inflated log Gaussian or zero-inflated Gaussian, has excessively high type 1 error. Monocle in this case works out. I think it uses some Tobit model in this case, which seems okay. And the quantile-based approaches, including zinc, are all fine over here in terms of robustness, in terms of protecting type 1 error. This is power. Over here are zinc. The two at the end are zinc over here. And you can see that they seem to lose power relative to some, but that's only because these guys have terrible type 1 error in the first place. So it's not even a fair comparison. We apply this to the CARDIA data. These are the numbers of things that are called significant. And the interesting thing is that despite the fact that some of these can't protect type 1 error, they actually find fewer things as significant over here. Versus zinc with the Cauchy tends to find a little bit more, regardless of this scale over here. In terms of overlap of what features are discovered, over here, we only compare the ones that have good type 1 error control. Blue is the zinc, all right? Over here, LDM, limma, and linear regression basically find the same things. And over here is a quantile approach that doesn't take QRank, which doesn't take into account zero inflation. So zinc is finding most of these things and a little bit more in this case. If you look at the cumulative sum scaling data, similar story. Zinc is finding most everything that everything else does, maybe a few that it doesn't, and quite a bit more over here. All right, so now let's look at the patterns of abundance profiles of taxa that are only identified by zinc. So the main thing that we found is that zinc actually discovers heterogeneous effects that are beyond simple mean effects over here because it is quantile based. So what we have here, the solid lines correspond to the quantile functions for the patients who have hypertension, the red for the non-hypertensive patients over here. And so you can see that even at the median, they're pretty similar, and that the split over here really happens later in the distributions over here. Okay, and so you see that if you're only looking at measures of central tendency, it's not really good. Doesn't necessarily look that different. But if you look towards the tails, you start really seeing an effect over here. If you look at this other example, which is looking at Haemophilus, the story is even more interesting in the sense that there's crossing over of these two quantile functions over here. So if you're looking at lower quantiles, all right, you can see that the function for the non-hypertensive patients is actually higher. But if you're looking at higher quantiles, the story is flipped. If you're looking at the exact median, you don't see any difference in the medians over here. And so when we showed this to our collaborators, they said this actually makes sense. The reason is that Haemophilus is actually a very heterogeneous genus, which contains a lot of sub-members. And so for some taxa, they are actually increasing risk of hypertension versus other taxa within the same group actually decrease it. So it's not just what's there, it's which specific members of this genus that are there. All right, and skip this. So just a couple of remarks on this point. All right, in general, reliable testing is still challenging, and there are no consensuses as to whether as to what are the best approaches. Every lab uses their own things. Many of the approaches out there are statistically totally invalid from the perspective of type 1 error control. All right, zinc is an approach that we think works reasonably well. It's robust to distributional issues, and it works under different normalizations. It has much better type 1 error control than many of the tailored approaches, and it tends to be more powerful than, say, linear regression and other classical statistical tests. The downside, of course, is that it does have many downsides. It doesn't work well for low-frequency taxa. That's simply the case. But the truth is also that the vast majority of methods don't work in this case. And when your sample size is very small, zinc can be very conservative. So it really is designed for the setting where we have large sample sizes and much, with these modern studies over here. All right, with that, I'm just going to conclude. Much of this work was driven by my former postdoc, Wodan Ling, who's now on the faculty at Cornell. I have a huge bunch of statistical and applied collaborators on this, and those are my funding sources. And I'm happy to take any questions now. Thank you.
As a first go on,
Oh.
I think there's a three.
What do I do with this? Oh, that's. Sorry, I closed that because it was.
Yeah. So
how do we get back to there?
Yeah, I know idea. Escape.
Yeah. Oh, there we go.
Yeah, okay.
Oh, chat.
Okay. That's.
So let's start from the top. So the question here, in certain multi-center trials, people use different instruments to analyze the same samples. Does Conquer handle that variability? And that's what we saw with the multiple studies. All right, totally different instruments, and in fact, different collection protocols. And Conquer is not perfect, but it tends to do much better than not doing any correction at all over here. So absolutely there.
Okay, let's alternate. Any question from the room?
First one, Mr. Barney, will you talk about batch effect? Do you assume they are also global?
Excellent question. So the question is the batch, do we make assumptions regarding the observability of the batch effect? Yes, we have to assume that we know the potential batch variable in this case. And that's how most of these batch correction approaches work. The ones that don't are typically people use surrogate variable analysis over here, which is Jeff Leake's approach, where basically they try to identify unmodeled heterogeneity, which they then say is some unobserved confounders or batch variables. Wodan and some other people, some of my other former students are working on this. I'm not involved because, well, they need papers that don't have my name on it for tenure. And so they are pushing that forward. I think their paper may be under review at Nature Methods right now, but I'm not involved in it, but they have been pushing it forward. It is a lot harder once you deal with non-normal data though, because these distributions are just you don't have enough structure on there to determine how many unmodeled batches there are. So it's an interesting question, but yeah, fantastic question here. Thank you. So the question is whether or not we accidentally remove treatment effect. In principle, it could happen, but here we're including the treatment effect in our batch correction. That's why I said we're double dipping a little bit, because we're using that knowledge already as we do our batch correction. So it should preserve it, but I'm sure there are some situations where you can show differently over here. In fact, we probably preserve some of the batch effect relative to the treatment effect in this case. So
my last question is for you also talk about the large scale analysis. So can we, in that case, can we treat or can we model batch effect into dispersion, like this unobservable? So it's got given a large number of sample sizes. Maybe to think about batch effect as a dispersion.
yeah. So you absolutely can put batch effect into dispersion. In fact, I think methods such as corncob and others try to explicitly, they don't deal with batch, but they deal with variables as driving dispersion versus other types of mean effects over there. But the reality is that, you're still only correcting for a couple of different moments where there could be higher order effects over here. So that's why we still prefer doing it fully quantile based, as opposed to picking some sort of quasi likelihood or something like that. Yeah. Thank you.
Let's go to the.
All right. Sorry, I'm gonna full screen this just so that I can see a little bit better. Naively, it feels like there's a connection between your emphasis on quantile based methods and ordinal logistic regression methods. Any thoughts? I'm not entirely sure I see the connections so much. I'm sure they are there, but my brain is not smart enough to see them. It may be obvious to others. I mean, my understanding of ordinal logistic models is basically those types of adjacent category logits and things like that. And I'm not entirely sure I see the connection in this case. Perhaps at a fine enough grid of quantiles, it may coincide, but here our data are basically almost continuous. After they are counts, but they're almost continuous with regard to their sort of dynamic range. So I'm not sure that that's exactly the same thing, but I'm willing to acknowledge I don't know enough about that and I haven't thought carefully enough about that to really say.
But may I make a comment here?
Yeah.
I think for the second project, the quantile regression essentially captures some kind of latent interaction.
Yes.
So I think it's kind of fundamentally would be different from the logistic model.
Yes, in some sense. Yeah. I mean, the one other thing I will say about zero inflation over here is that it overemphasizes the difference between 0 and say 1. And that is another limitation of the approach. Versus, as you said, it's latent. In truth, it may be truly continuous, but it's just we've artificially created this dichotomy over here. So, all right.
Yeah, any question? Laura?
Yeah, I want to know if the zeros that two zeros or just values that special even with the foreign assembly versus values.
So the truth is, so the question is whether or not those zeros are true zeros or if they're just because we have insufficient library size, basically sequencing depth. And the truth is nobody knows for sure. And depending on who you talk with, their opinion is different. We've chosen in this case to model it separately because we believe that many of these are true zeros, like some bacterial taxa just are not in certain samples. But it's impossible to say without the contextual knowledge. If you truly believe that that's just some sort of detection limit, then you probably don't want to use these zero-inflated models because you are so overemphasizing this 0 versus a tiny number. Like a tiny positive number is basically 0 in linear regression, but here I'm treating them as totally different in a logistic model. So it's a good question, and I don't know the answer, and it really depends on who you're talking with in terms of my collaborators as to whether or not they believe they're true zeros versus detection issues. right? So this one, any chance you're removing population differences with the batch effect? So I think that's similar to the previous, to the other question over there, where, you know, that is absolutely a possibility. We try to preserve it in our modeling, but there may be situations where that falls out over here. Okay.
Just a follow-up on that.
Yeah.
So is it possible to partition the variabilities that say some portion of it is truly the batch effect you want to hammer out, and some of the variabilities is actually the differences you want to celebrate? Because the more you do mega analysis, the more those variabilities is going to show in your finding.
Yeah, so the question is whether or not I can partition that up front. And I think that's a hard problem. I'm not sure. I think you're asking too much of your data in front of you in order to really pull that apart. There may be a way to do it, but I don't see it as particularly obvious, because that's the entire problem here, is that I don't know where it's coming from. And so I choose to preserve all of the variability from the hospital or treatment effect up front. But the fact that I don't see it doesn't mean it's impossible. It's just I don't know how to do it right now. And it seems like a case where you're asking a lot from your data. Because if you knew that, then I probably wouldn't have to do much analysis up front to begin with.
But I guess even you remove upfront incorrectly, but if this is not the primary covariate, so the question still remains whether you decrease the power of your primary kind of covariates or not. Probably not, unless this covariate is dependent on the primary covariate.
Yes. So you basically have to adjust for anything that you think is influencing batch, and any variables that you think are interesting and important. And so those guys will be preserved, especially your primary covariate of interest. You're preserving the effect there. If anything, our type 1 error is theoretically inflated over here. So your observed power is higher than it's supposed to be. But if you have something else in there, some other covariate that you don't have, you could lose something there in principle, because you've over-adjusted out certain effects. That is true. That's a great point. Thank you. Exactly advocated for continuous data.
It was, the talk is recorded.
If the method is not very good for rare data taxa, how rare would be the limit? Microbiomes can have huge diversity, which means even the most abundant data have a lot of zeros and low counts. So rare is an operational word. It's much like when we deal with rare genetic variants. It's purely operational. If your sample size is 100,000, all right, then a prevalence of 1%, you're still going to see it in 1,000 people, and so you're still going to be able to use it. But if your sample size is 100 and it's as a prevalence of 1%, you only have one person where this taxon is observed. You're not going to be able to do anything there. So it's really operational. I don't have a specific guideline other than you kind of know when you see it. And that's oftentimes how we do a lot of our data cleaning in our experiments, right? If our sample size is really large, then we'll keep anything in there that has that shows up in at least 1% of samples. If your data set is really small, only 30, then you might restrict to taxa that are only present in 30 or 40% of samples, or maybe even higher in some cases. So I'm sorry, I don't have a specific answer. It's very operational. Right. Questions in the room?
Questions?
The tools, if you email me, I can send you to the GitHub sites for these tools. They're all freely available. I know the Conquer approach, several groups are already using it. The Zinc approach, I'm trying to convince more people besides me to use it. So hence why I gave this presentation. Survey sampling methods, batches as random clusters.
When you go through the literature, if anybody's using survey sampling methods at all. I know when we adjust for site effects, for example, so we can use survey sampling methods, thinking of the sites as sampled from a random sample of sites going to a thinking in a meta sense, and it works. So I wonder if people can think of batches as a random sample from the batches and take that kind of approach, and think of them as exchangeable.
Yeah, that's an interesting thing. Actually, I think in some of the early gene expression work, I mean, some of it is empirical based, and so there's a natural connection up there already. And then some of it, I think I have seen people trying to model them as random, but I have not seen that work really become prevalent. Some of it is because a lot of it tends to be on adjusting for batch effects up in the downstream analyses, rather than trying to remove it up front. So I'm sure there's more work on there that I'm not aware of, but
probably not enough, because
yeah.
that's the key, trying to remove it up front, right?
Yeah, but I'm not sure beyond that. So yeah.
One last question, Julian.
So in one part, it's like you should be using, instead of grids,
Yes.
it's multiple like a quantile regression model.
Yes.
Is it gonna be more prone to overfitting the data?
It's
Compared to like other methods, say combat-seq or like just combat, they're just using one single like model.
Yeah, so the reason why we need to look across a fine grid of quantiles is because we're not willing to assume a particular distribution. If you're, the thing is, if you have a normal distribution, then all you need is your mean and variance, and that fully characterizes the entire distribution. Versus here, I need to characterize it at each grid. So I guess in principle, it could lead to some more variability in some of the estimation, particularly for the lower and higher quantiles. But I don't think it's that different than if you index it. I still think it's probably more advantageous than indexing it with just a couple of parameters like a mean and variance.
Great. Let's thank Dr. Wu again for the very stimulating talk.
Okay. Thank you.